Can gene editing change cystic fibrosis carrier status? Cystic fibrosis is a genetic condition caused by mutations in the cystic fibrosis transmembrane conductance regulator gene, often shortened to CFTR. Individuals who carry one mutated copy of this gene are known as carriers. They usually do not show symptoms of the disease but can pass the mutation to their children. Gene editing technologies, such as CRISPR and base editors, are being researched to correct CFTR mutations. These methods aim to restore normal function in cells affected by cystic fibrosis. Current studies show that gene editing can successfully target airway epithelial cells, which are primarily impacted in cystic fibrosis. For instance, researchers have used base editor complexes delivered via aerosol to animal models, achieving lasting corrections of CFTR mutations in airway cells for at least 12 months. Additionally, these techniques have restored CFTR protein function in human airway cells grown in the lab. However, it is important to note that these gene editing methods are still in the experimental stage. They face challenges, such as effectively delivering the treatment to all necessary cells, ensuring long-term safety, and achieving comprehensive corrections across different tissues. When it comes to carrier status, gene editing could theoretically change a person's CFTR gene from a mutated to a normal sequence in their body cells. This change might prevent disease symptoms if done early and thoroughly. However, carrier status is determined by the presence of a CFTR mutation in germline DNA, which is the genetic material passed down through eggs or sperm. Current gene editing techniques focus on somatic cells, meaning they do not alter germline DNA. Therefore, these methods cannot change an individual's inherited carrier status. Germline editing, which would involve altering the DNA in eggs or sperm, raises significant ethical, safety, and regulatory concerns. As of now, it is not practiced in humans. In summary, while gene editing holds great promise for treating cystic fibrosis by correcting mutations in affected tissues, it does not currently change a person's inherited carrier status. Carrier status is a genetic fact based on inherited mutations, while gene editing therapies aim to correct mutations in body cells to alleviate or prevent symptoms. Ongoing research and clinical trials are essential to develop safe and effective gene editing treatments for cystic fibrosis. However, altering carrier status through germline editing remains a theoretical concept and is ethically complex. Individuals who have concerns about carrier status should seek genetic counseling and professional medical advice for family planning and disease management.